So I have a full semester of Bio 2 and I have a mini semester of Bio 2. You guys are in the mini semester, okay? At the beginning of the mini semester, not a big deal as far as my confusion factor, but as the semester has gone on, guess what happened? Y'all have come very close to in line as far as what we're talking about. So sometimes I forget if I mention something in class and I go, remember we talked about that the other day, remind me, because I did talk about it the other day, but it was in the other class and it's getting difficult. So I can't remember if I said this. Chapter 31 on the fungus, we did not do a lab on that. Right, yes, that's right. We did not do a lab on that. We're not going to do a lab on that. It's not going to be on the lab practical. I'm not going to go over it in the lecture. It's not going to be on the final, okay? So even though, with all due respect to the fungus, it just becomes a time thing at this particular point. I just want to make sure that I have enough time to go through the animals. Not to say that the fungus aren't interesting. And if you're looking for some summer reading, that's not a bad chapter to read. There are some. They have some. Fungus has some very different ideas about how this unity or this thing of life. You remember Bio 1, we said that the first entity that has all the characteristics of life is the cell. Fungi go a different route from that. So and again, I don't, I'm not going to talk about that. But if you do want to find some interesting ways of being alive, you can talk about or read about the fungus. So nonetheless, for your last exam. Your next exam Monday, which is the plants 29 and 30, for your last exam it'll just be animals 32 to 34, which we do 32 today, okay? All right, so hopefully there's no mistakes about that, but I guess the bottom line is, the summary is, there's nothing about fungus on any of your exams, lab or lecture, but there is going to be something on animals, and you betcha there's going to be something on animals, and not these guys. See that little record album there? I always put that up there. I think that's kind of funny, because to me, I go, oh yeah, that's a record album, but I wonder, the millennials or whoever, if they look at that and they recognize that as a big, the big black record album. And I thought, well, wait a minute, CDs kind of look like that too, but you may not remember this group, the Animals. Right, and House of the Rising Sun, you may not remember that song either, but if you heard it, you might have. I put it on there. I always find it kind of funny that these guys, look at the way, like the Beatles when they first started out, and these musicians from back in the Animals must have been the early 60s, I'm sure. They look very, you could, hi mom, this is my boyfriend, there'd be no problem. But these are rock and roll musicians. That behind the scenes, they're probably not all that clean cut, right? So. And then as time went on, they didn't care about how they looked. But anyway, I think that's interesting. In the background is one of my favorite critters, obviously. I love fish. I love snakes. I'll say it a million times. I have a lot of different favorite critters. But I've got a large picture, obviously, of snake scales. And to me, it's fascinating how it works, kind of like, pixels on a computer camera. Each little scale has a color, or even sometimes a shading of color. And then you put those scales together and you get a pattern. I think that's just absolutely magnificent. And of course, if you think about what we've been talking about all semester long, and the fact, let me turn my camera on so you can see me shaking my hands all around. Yeah, there we go. All right, anyway, so if you think about all of those scales put together through random variations being selected for, all that evolution stuff, to me, that's even more overwhelming, Folks say, what are you passionate about? And to me, I always like to answer, just because everything is so incredibly complex, yet it came together randomly, right? I mean, we talked about that in evolution. If you're born with a genetic advantage because of the way crossing over occurred, and that's typically how it works in us, not through mutation, right? But through the way your chromosomes, the way your parents' chromosomes crossed over meiosis, which is all due to probability, that's kind of overwhelming, right? To think about all this diversity we have on Earth, and to think that it just kind of came about through this trial and error process of evolution, all right? But as we've done all semester long, especially back when we were defining what the heck even a species was, let's talk about what makes animals animals, all right? So we talked about what makes a plant, and we didn't talk about fungus, but anyway. Prokaryotes and protozoans. Protozoans, that was a little bit weird. Yeah, I get it, because there's a lot of different things about them. other than they're small. Remember, that's why we used to put it in Protista. But let's talk about what makes an animal an animal. All right, the bear standing there in the stream. He's obviously waiting for the salmon to swim upstream. If nothing else illustrates this whole randomness of life, as you're thinking about the salmon is swimming upstream to reproduce in order to engage in one of its living characteristics. And of course, the bear's got something totally different in mind, because the bear, like you and I, is what we call a chemoheterotroph, and that's not unique, but that's found as a general feature of all animals. All right, chemoheterotroph. Now, the hetero means other, and what we mean the other is where we get our carbon. Living things, all living things on Earth anyway, need two very important substances. They need carbon, because we're a carbon-based life form, and energy by which to put that together, okay? So the hetero means that we, us heterotrophs, we get our carbon from another organic material, food. We're not plants. Plants would be autotrophs. They can make organic food out of stuff that's not food, carbon dioxide, okay? And we do that chemically, because remember back in the prokaryotes, there were some that were actually able to use the sun. I think it was, if I remember correctly, it was the purple bacteria that's actually a photoheterotroph, okay? But we are uniquely chemoheterotrophs. We get our carbon from another organic compound, okay, to make our molecules that we need for us, right? And we get our energy from that as well. We don't get it from the sun. Multicellularity. Now, this is something that this is something, oh gosh, it's between the rungs of Aristotle's ladder again. All right, what is multicellularity? What makes something that's unicellular multicellular? Is it two bacterium when they come together, right? In a very simple streptococci or diplococci, I should say. Is that multicellularity? Well, yeah, by English, right? Multicellularity. When the cells come together and form the biofilms in the prokaryotes, or when the cells came together in the ball box and they form the colonies, and we talked about this in lab, is that multicellularity? Yes, that's multicellularity by the English definition of it. But here's where I'm going to kind of go into the gray areas, the parts in between the rungs of the ladder. Some animals they're multicellular, but we call them more precisely a loose aggregate of cells. A loose aggregate would be, oh gosh, right before a tight aggregate. You know what an aggregate is? A collection, okay? Now, specifically, we're talking about the sponges, and we'll talk about the sponges in more detail later on. But a sponge is an animal, believe it or not. I remember that was one of the neat things I found out when I was getting into biology. Like, oh my gosh, I didn't realize that. A sponge is an animal. It is multicellular, but the cells are really more on the loose aggregate side of multicellular. Now, to be fair, when does a biofilm or a colony become a loose aggregate? When does a loose aggregate become a multicellular? Somewhere along there, right? Everything's not black and white. Now, the reason why I'm spending so much time emphasizing that is because one of the general features of animals, and one of the, actually, should we say, the innovative features of animals, or one of the things that the evolutionary pattern of animals came up with, was this idea of tissues, okay? And sometimes we refer to plant tissues. And again, this is kind of a definition thing. In the technical zoological definition of a tissue, we think of an animal tissue, all right? And the reason why we do is tissues are like multicellularity one step further, all right? If you think, and again, it's not really a step, it's a blending, right? So we get this single cell, biofilm maybe. And don't think of this as an evolutionary thing. Just think of this as trends we see in organisms. Colonies of cells. In sponges, loose aggregate of cells. And now what animals have done is taken that multicellularity and they form it into what we call tissues, okay? Now, don't worry about what they're all but peripheral. I'll get that in a second. What is a tissue? Take a look at this picture right here on the left. Or on the left. Holy cow, am I facing the wrong way? On the right. This is actually a picture of what we call the neuromuscular junction. Now, I don't know if y'all have taken A&P or a neuromuscular junction, but I guess if you look at it, you can figure out what I'm talking about. This is the spot where that long wire thing, that nerve, stimulates the muscle. That's muscle cells down below and what we would say muscle tissue. And above that is nervous tissue. You see, the reasons why tissues are a big thing in animals is because tissues allow these very complex things that animals can do. Like, for example, jumping down below for just a second to complex movements, since we're talking about muscle cells and nerves. So tissues are a group, they're multicellular groups, okay, beyond a loose aggregate, but they're structural, functional units. Nervous tissue conducts nervous impulses. Muscular tissue contracts, moves joints around. Digestive tissue, endocrine tissue, when you take AMP, if you haven't yet already, was it AMP1, I think? They talk a lot about tissues. We have adipose tissue, and then within tissue, there are tissue types. It's all about tissues with the animals. So that's why, I got to get you back to Aristotle and understand that, when does it, all but peripheral. Sponges are peripheral. They're very peripheral. They're very perforated holes, right? Peripheral. The scientific name actually makes sense, doesn't it? Anyway, why aren't they tissues? Well, because they're not quite tissues, right? I suppose you could argue that maybe sponges do have tissues. I don't know if too many zoologists would agree with you, but considering the technicalities behind it. But again, the whole thing about it is we get these groups of cells that have these very special functions, right? Nervous cells. I've talked all about that, all right? Does that make sense? Does that make sense? That whole jumping from a single cell to, just two bacteria and all of that, and how, when does that become a group of cells that are living together just as a colony or as a biofilm, like they were in prokaryotes? When did they become these structural units? Because, of course, if you remember in the Bio 1, the hierarchy beyond tissues is tissues make up organs, organs make up organ systems, and right? Okay, we will talk about tissues again later on, obviously, because this is so important. One of the things that animals don't have, you learned in Bio 1, is we don't have cell walls. Don't need them. Don't need them. Got a skeleton. Got one on the outside, maybe. An exoskeleton. Some critters have that. Endoskeleton. Some critters like worms have what we call a hydrostatic skeleton. Hydrostatic, water. When you put your thumb over the garden hose and the water is flowing, you know how the hose gets stiff behind it? That's kind of what we mean. Earthworms, that's how they move. If you ever watch an earthworm move, it kind of looks like what it's doing, like waves of pressure being moved along as the animal moves along. Now, without cell walls, that means we do have to have some type of a skeleton. Plants have their cell walls because they don't have a skeleton, so they use their cell walls to be able to stand up against the force of gravity. Now, we have a skeleton by which to do that. Okay, and then we have the added feature, because we don't have cell walls, we're flexible, right? Now think about that. If you think about do plants move? Yeah, the leaves kind of turn into the sun. Do fungi move? Well, they kind of grow into things. Do prokaryotes move? Yeah, they do. But we're capable, like the next one says, of complex movements, swimming, flying, walking, grabbing, touching, jumping, whatever the case may be, right? Which means diversity, which means that animals, because they don't have cell walls and because they have these functional units, can engage in things like that butterfly right there. You don't know any plants that fly, do you? I know a lot that drift in the pollen or the seeds, but I don't know too many. I don't know of any plants that walk. I don't know of any fungus that there's some that creep kind of weirdly like, but there's none really that walk, right? So that's unique among animals. We have this ability to have not just turn toward the sun kind of thing, right? Not just use our little flagella to make our way around kind of randomly in this drop of water, but we can do really complex things, can't we? Now, there are those that are sessile, there are forms, or sedentary forms, and in many cases with animals, this is very cool, because this is like what we call ecosystem evolution. In many cases with animals, think about the butterfly there, a young butterfly, or before it's a butterfly, it's a caterpillar, right? So in many cases of animals, if there are sedentary forms, sometimes they're mixed in with mobile forms. So sometimes, like say the adult will be the sessile form, the one that stays in place. Think of like coral, okay? And then the larvae of coral can actually drift. That dispersal. You want to disperse. We talked about that with plants a bunch of times. Even in animals, disperse, get out, exploit other resources, invade other environments, get away from me, don't take my resources kind of thing, all right? And that's all, again, because of this ability to move around complex. You can either drift in the water like the larvae of the coral do, or you can walk or fly or swim, you know, whatever trips your trigger, whatever environment you're in. Then all that ties in, and that's why we have so many different kinds of animals. Over 8 million species, 35 to 40 phyla, and of course my favorite, right, would be the fact that most of them are in the ocean, because that's what I love the most, right? All kinds of cool things like millipedes you see in that picture, weird looking things that look like a sea star and are related to sea stars, but actually called brittle stars. Now they can walk, very bizarre. Even on land, later on when we get to that part, I'll show you a brittle star walking on land. There is the chameleon with this incredible tongue, and not only the fact that the tongue can reach out and grab an insect, but the eyesight, the visual acuity, the reflexes to be able to snatch that sucker out of thin air is amazing to me. Sexual reproduction, yes, that's what those tortoises are doing. I always put that up there as a remembrance. I was at the San Diego Zoo years ago when I was in the Navy, and went by the Galapagos tortoise exhibit, and there was half dozen, 8, 9, 10 people there talking, and they were, what is that? Are they aggressing? Are they fighting? What are they doing? And I'm like, oh my God. Anyway, so yes, that's what it looks like. It's not particularly fast. It may not be particularly exciting to watch, but for the tortoise, it must provide some excitement. Here's some complex movement. This is one of my favorite stories, the albatross. They say that male albatrosses, once they're born, never touch land again. Now, I don't know if never is the right word, but they rarely do. And you're thinking, what do you mean? Well, they feed on fish, so they may dive into the water, and they mate. This is kind of the cool part. They mate in the air. So females would land to lay the eggs, but males would have no reason to. Now, again, I can't imagine a male, if he was tired or needed to, would land on an ice floe or something like that. But they can float in the water too, you know, like ducks do. So we got sexual reproduction, which is genetic mixing. Remember, in most critters, that whole crossing over and meiosis is where most of this variability comes from. In most critters beyond like the prokaryotes who could mutate, because they don't do meiosis. There's no alternation of generation. That's kind of cool and a very big difference. So that's a very big change that occurred in that branch, that bush of life, huh? Alternation of generation. It worked really well with the plants, with the gametophyte. Right? The gametophyte was getting smaller, wasn't it? It was shrinking. The gametophyte is haploid. And although that works very well for mosses, okay, and that kind of stuff, when you're moving out into the world and you're haploid, it's nice to be diploid. You can be haploid. That's why they're haploid critters today. Don't ever think that one's better than the other. It just depends on where you live. But diploid has a double set of genetic information, and we talked about that in evolution. You got those hidden alleles. You got more genetic ability to deal with changes in the environment, okay? So in the case of animals, what you're basically doing is, without the alternation of generation, is you're very much protecting that gamete, right? Our gametes are produced very much on the inside of us, right? And not only that, you're really giving a lot of care in many cases to the offspring. Not in all cases. Many reptiles lay eggs and that's it. But now we have evidence to suggest that even dinosaurs, there was maternal care. That female dinosaurs, maybe even males, because in birds, which are dinosaurs basically, there are some males that take care of it. So, you know, less emphasis on numbers, more, it's a quantity quality thing. If you take a lot more care of your young and protect the gametes, then you don't have to make as many, okay? Down below is a picture of an animal embryo. It's probably a sea urchin, since that's what they do most research in. And those little lumps are indeed cells. And this is what we call, looks like the 8-cell stage. I can't see any cells below it or above it, right? So it looks like we got one cell, the zygote, into 2, into 4. So this is in the 3rd division, right at the beginning. Animals have very unique embryonic development, all right? We'll talk more about that later on, but that is another one of those, what you would call hallmarks or milestones of being an animal. Let's return to multicellularity again. Not that this is the last time you'll see it, because I'm going to keep talking about it. It's very important. But multicellularity, I read an interesting paper, and it said that in order for cells to achieve true multicellularity, so we're not talking about loose aggregates and colonies and all that sort of stuff. You have to stick together. Okay, all right, well, that's something other cells do. But they have to signal each other, and other groups of cells may do that. But they also have to work cooperatively, and they have to work cooperatively to a common function. They have to specialize. I thought that was kind of neat. And the reason why that's kind of neat is, if you remember back in the protozoans, and if you don't, I brought it forward so you can, there was a protozoan that we knew as the choanoflagellates, okay? And it was a protozoan that very often existed as a colony, like you see in the picture there. And on the right is an individual choanoflagellate, an individual organism. And on the right on the picture in the black and white, I kind of show you what those might have looked like. So an individual choanoflagellate binding together with a bunch of other. Now, choanoflagellates are what we call collar cells, because around their collar, around here, there are some it might be cilia. I'm not sure what that is. But anyway, the flagella and the cilia are used to capture food. So maybe being together like that with all of your flagella, like you see in the picture on the right, made you more efficient at turning over more water. So these guys are capturing food in the water, and the more together you have, the more water you can turn over, because you got more flagella kicking water around, right? Okay. Now, take that. Those are protozoans in the black and white, all right? Those are drawings of protozoans. Now, jump down below. We haven't talked about them yet, but this is a microscopic picture of cells that we find in sponges that we know as choanocytes. Now, they're not protozoans. Sponges are not protozoans. They're animals, right? Another evidence of how messed up the protozoans are and how paraphyletic they are, right? Here is a protozoan that's more closely related to animals than it is to other eukaryotes, other protozoans. Now, I talked a lot about that in protozoans, so I'm not going to spend any more time on that. But look at those cells. Don't they look similar? I mean, isn't that kind of a whoa kind of moment? It is for me. I look at choanoflagellates that are protozoans, and I looked at these cells that we find in sponges, and we'll talk about them later on, and I say, wow, those look alike. And oh, by the way, when we get into flatworms and other critters, we're going to be introduced to these things called flame cells. Now, what flame cells do, nothing to do with fire, has to do with excreting waste, but how flame cells look and the way they do it looks a lot like choanocytes, looks a lot like choanoflagellates. Isn't that kind of a neat connection between those two? That's kind of that rung between the rung with that Aristotle. Also, when we look down at the genetic level, there's a lot of similarities between the genes that code for the things that cause those choanoflagellates to stick together. Proteins, specifically collagen is one of them. Collagen, which is one of those proteins you talked about back in Bio 1, is a very important protein found in animals, and it helps, among other things, to stick cells together. So we find that gene that codes for that in choanoflagellates and animals. So we got genetic evidence. So we got the very simple, morphological, oh, they look alike, right? And we got some genetic evidence, because remember, similar proteins, similar amino acids, similar genetic code, right? So here we are again. Pull back, and there we are. Yep, one more time. Doggone you, protozoans, you're everywhere, right? So we're more closely related to a protozoan than we are to anything else that's in our group beyond the protozoans, the fungus, the plants, and that sort of stuff. All right, now another very innovative thing that animals came up with are these things called homeotic genes. And these pictures, the one on the right especially, I pull up the web, can look a little complicated. But basically what we're talking about in the colors, the one on the left, the little critter there with looks like fruit stripe gum to me. I always think of fruit stripe gum when I see that. But what the different coloring is talking about is the influence of different genes. genes. And you can see down below in the one on the left, they got the gene that they've called the antennapedia, the eulothorax. They got genes that control different areas of the body. All right, that's what homeotic genes are. Homeotic genes are genes that control different areas of the body. In other words, what I mean, where things are found, where you put body parts, where you put the head, where you put the legs, where you put the wings, where you put the different things. Now, animals have homeotic genes, but so do plants and so do fungi. It's how plants know to put a leaf here, a root there, a cone there. It's how fungi know to build a spore-producing structure here. It's basically the information on how construction, how you put this thing together. But in animals, all right, take that to the next level. Not that other critters don't have it, but take it to the next level. Because in animals, what we get sometimes, and also in plants and fungi, we get a sequence of homeotic genes, which we call a homeobox. So in the right, the different colored genes in the Drosophila, that's a fruit fly, by the way, the light green, the green all the way to the yellow, that's what we call a homeobox. There is a sequence of these placement genes. Now, regulatory, so we're talking about genes that control it. She did for many years until enough speeding tickets changed her behavior. But I kept trying to remind her, Stephanie, the accelerator is variable. It doesn't have to be full blast or off. There's things in between, okay? So these Hox genes are kind of like that, is what I'm trying to say. That with regulatory genes, what you're doing is you're regulating when something starts. And since these genes are very associated in the embryo, so you're starting early. When does this body part being placed here start? How long does that happen? How long does that body part grow? So growing an arm, right? You grow. When do you start growing the arm? Where do you grow the arm? Well, you ought to grow it off the shoulder. How long do you grow the arm? Well, you grow the arm, whatever evolution says is a good length. Or how long do you grow the leg or the flipper? Or whatever else that not mammals, but animals have, right? So any changes in those genes, any duplications, any shuffling that occurs through evolutionary time is going to end up in many different things, much variation, morphological diversity. That's the words I'm looking for. So if you look on the left, what we're saying is, whereas those genes at one time were all kind of mixed together, the fruit stripe part, the green, the yellow, is that orange maybe, red, when they were all mixed together like that, or what we get is a critter that has the same appendages over and over and over again. So it's like repeat. Now, there are critters that are still alive that are that way, but what we're saying is what could have happened in the evolution, and we're just kind of looking in this case among the arthropods, the insects and their relatives, if those genes begin to separate out, if they weren't all fruit striped in, then we might get specialization. We get a green part with a thorax where there are some walking legs, and we get the head part where are the eyes and the sensory organs and the antenna, and then we get a part that has these big jumping legs, and this is just in the grasshopper. And then in the abdomen, we would have like the digestive parts, maybe the reproductive parts. And then, actually, no, I take that back. The blue part is the reproductive parts, the genital segments, okay? It's what we call segmentation. And we say that in evolution, that was an important change from kind of mixing it all up and just kind of having a bunch of appendages to having very specialized appendages, walking appendages and jumping appendages and different segments. Right, segmentation is another characteristic of animals that's very important. And so now you can look at the fruit fly, and we say, all right, they had positional genes. They had these regulatory genes that said where you put things and when you put them there. And maybe what happened, or one of the hypotheses that we suggested in the case of the animals, specifically humans, is duplications of those genes, sequences of the genes, shuffling them around, putting one gene next to the other, which increases the effect, putting one gene next to the other, which decreases an effect. But all of those changes, and as we move down the picture, you see now in the human being, you've got the blue areas, the green areas, the orange areas, and those genes are controlling different things. They're controlling where different parts are. Now, the important thing about all this homeotic genes and homeoboxes, if you got a lot of ways to regulate when and where you put body parts, you can have lots of variation. Think about, trying to get a good analogy, think about controlling, think about my daughter again. If she understood, which she eventually did, that you just barely push on the gas pedal, you go slow. push on it halfway, you go a little faster. Think about that's the same way. You got many different ways to get from point A to B in a car using the gas pedal. Really quick, really slow, and everywhere in between. It's almost infinite. It's kind of the same thing with these homeotic genes. You got many different ways to control where you're putting body parts. So those many different ways allow you to have many different variations of ways. I read an interesting article about why there's so many different breeds of dogs and how that happened so quickly, because 10, 12,000 years ago, humans began to domesticate wolves. Genes that control the way they, essentially the way they look, their fur, their height, their faces. So as humans, through artificial selection, we've exploited that. Mix this one and this one and this one, and we get this one, mix this one and this one. I think about Cooper. Cooper is a Cavalier King Charles Spaniel. He's not even really a Spaniel. Cooper is a reflection, or Cavalier King Charles, is we looked at old paintings of these English folks, and it might have been King Charles. They had so many of them. And he had this dog, and we liked the way the dog looked. We didn't know what breed it was. It seemed that the breed had vanished, or whatever the case may be. And we got together, we put dogs together until we made one that looked like that. Cooper and the Cavalier King Charles is very closely related to a Chinese breed of dog known as the Chin, the Chinese Chin. And a relative of Millie has one of those, and the fur, he doesn't look like that, but the fur is identical. It's funny. You see what I mean? Diversity, right? That's why we get so many different animals. That's why we get, over 8 million different species of animals, and they're finding new ones all the time. Okay, let's go back and talk a little bit more about embryonic development, because that's all fitting in with this idea. Oh, too far. Because that's all fitting with this idea of genes. And let me start you with what you remember. You remember the zygote, right? So that's the first cell. That's the sperm and the egg come together and starts to divide. It undergoes cytokinesis, right? Just like we talk about. So the arrow of cleavage, that's all mitosis, right? So we get the 8-cell stage, like that picture I showed you. And that keeps growing into a ball of cells that, before it becomes a blastula, becomes this really cool thing called a morula. And a morula is really cool in the sense because where that word comes from. It's Latin for grapes. Okay, so it's a group of grapes. So it's kind of like Staphyla is a group of cocci. A morula is a group of cells. And then that morula becomes a blastula. And the reason why that would differentiate is a morula is a lump of cells. A blastula is a hollow lump of cells, because you see inside of a blastula, there's a blastocele. Now, why is that going to be important? Well, it's going to become important because animals have body cavities, don't we? You have a body cavity, right? You have the body cavity that your lungs are in and your heart is in, the body cavity where your stomach and your spleen and all them other goodies down there. They're in a body cavity. You're not a solid mass of cells. You're groups of cells, tissues, more or less suspended in a body cavity, right? Some organisms that we're going to see have really extensive body cavities, and some don't, and some have repeated body cavities. Go back to your buddy the earthworm. Earthworms have body cavities that repeat themselves, so the emphasis here is cavity, okay? Now, what's going to happen in animals is when we have that cavity, we can start playing with the cells through these developmental genes and doing things with it. For example, cells can begin to enfold. It's like if you took a balloon and you push your finger into it, you didn't pop it, but you caused it to enfold, and that's called gastrulation. Now, that word gastrulation, if you picked up on it, gastrulation refers to the gastrointestinal. So we're talking about building the digestive system. We're talking about building the tube that it connects your mouth to your anus, right? Basically, not the planet. This is your digestive. The animal that we're going to dissect tomorrow, the worm that we're going to dissect tomorrow, is basically known as a tube within a tube body plan. At the very simplest form, this gastrulation ends up forming the digestive system. By the way, when it first starting, it's called an archenteron, which means primitive gut. All right, it's basically, if you looked at it this way, it's going to form a tube within a tube. That's what the yellow part. That's going to become the digestive system. That spot where you pushed your hole in the balloon, that's called the blastopore. So we say that the cells begin to turn into the blastopore, push in, and they form this arc enteron. Now, this is not finished because that yellow part is going to grow to the other end, so we get a tube within a tube. It's not going to cut it in half, remember, in a three-dimensional world. So it's like if you could push your finger through the balloon, and without popping it, you could open up, so there'd be you turn the balloon to a donut, okay? If that makes sense to you. Now, that means that what you're going to be able to do is you're going to be able to monkey with different kinds of tissue. And being able to do that means that you're going to be able to do different things because you're going to form like endoderm, ectoderm, and there's another one that's not listed there that's known as mesoderm. Now, mesoderm, these three things are called embryonic germ layers. Germ in this case doesn't refer to a prokaryote; it refers to give rise to. These are beginning tissues, and these are going to give rise. And we'll talk about this more in just a second. But these types of tissues: nervous tissue, digestive tissue, muscular tissue. Okay. So it's right at this point, very early on in embryonic development, we see this big departure from other critters out there or other organisms because what we're doing is we're specializing in tissue types. Okay. All right, now, in animals in terms of development, beyond just here at the cellular level, animals are very unique in cases because in many animals, or in most animals, there's more than one larval stage. All right, the larvae is a specialized definition. A larvae is something that's sexually mature. It doesn't even look like the adult, and sometimes it eats different things. So the caterpillar is a larvae. Okay, the caterpillar doesn't look like the butterfly, and more than one larval stage where you got the caterpillar, the chrysalis when it wraps itself up, and then before the butterfly comes out. And as I said before, one of the things that animals are very clever about when it comes to having these larval stages is what we call, this should be partitioning, not partitioning, it's spelled wrong, resource partitioning. Caterpillars eat different things than their adult butterfly counterparts. So they can live in the same area, because the caterpillars eat the leaves and the butterflies feed on the nectar that comes from the flower of the plant. Right? Everybody can live together. That's ecosystem evolution. Kind of neat. It's kind of symbiosis in a grand level, isn't it? And in a lot of cases, we have this metamorphosis that occurs. Like if you're looking at our buddy the mosquito down below, the metamorphosis then creates a juvenile. And the juvenile is a different term than a larvae. A juvenile is something that looks like the adult, but is still sexually mature. So in the case of humans, maybe you'd, I don't know, I guess when they get to be toddlers, they might, well, no, when they're juveniles, 8, 9, 10, 12. They're not sexually mature yet in the most cases, but they look like an adult. So in the case of the adult, or the mosquito down there, we get the eggs, the larvae, which does not look like the adult. That metamorphoses into a pupa, which doesn't look like the adult. And then we get small mosquitoes getting big mosquitoes, and they look like the adults, but they may be sexually mature. Probably not for very long, unfortunately, with mosquitoes. Okay, so some help. And again, we're going to come back to this because it's very important to understand how this fits into animal evolution. But I sort of want to go back in time again and remind you what we talked about in chapter 26, is that the one? So the evolution of life on Earth, and we're going to focus on animals. So going back before the Cambrian explosion, and that's at about 542 million years ago. You see right there, the Cambrian explosion is right there. We had some animals. The common ancestor has been around of animals for, what, 750 million years. So, but these animals were relatively simple. It wasn't until really about 560 million years ago we started getting bigger animals, macroscopic fossils. This is all during, these are what we call the Ediacaran animals. This is before the Cambrian explosion. And even a first macroscopic fossil, so smaller ones, right? But macroscopic fossils, you know, that we could start to see things in animals that we saw even in animals today, like radial symmetry. Right, there's radial symmetry among animals. You think about jellyfish and sea anemones. Now, not everybody's got radial symmetry. You and I have a different type of symmetry. We have what we call a bilateral symmetry, but we do see this idea of symmetry. And we'll talk a little more about symmetry in just a second. Segmentation. We saw in the animal line there was this evolutionary trend towards segmenting and specializing and having different body parts doing different things. And of course, as we move into the Cambrian, we talked about this before, what was driving this big one of the things that was driving this big evolutionary innovations was predation. Nothing drives evolution, well, I shouldn't say nothing, but evolution in terms of how fast natural selection, the speed of it, is driven very quickly by predation, because guess what? If you don't adapt, you go extinct. If you don't adapt to being a good predator or, surviving as a prey. And the reason why we know this was happening is we look at the fossil evidence. There's a borehole. See that? That's evidence of predation. Something was boring into this, whatever made this cast or was living in this cast and ate it. Or there are organisms that do that today. So we see this, this strive to remain alive. Now here it says 1.3 million species named to date. I think earlier I said over 8,000. I believe that's an estimate of species. So 1.3 million species today. That's still pretty impressive. All right, so we move out of these relatively simple types of critters to what Darwin had problems with during the Cambrian explosion, when it seemed like all of a sudden that we get these critters appearing all at once, right? So about 500, there it is, Cambrian explosion, 525 to 535. And a lot of that, again, had to do with this whole arms race, this predator-prey relationship. Now, when we go into the Cambrian explosion, and again, this is where Darwin had his problem. One of the things we notice about fossils from the Cambrian explosion have gone from radially symmetrical to having bilateral symmetry. Now, bilateral symmetry we have left, right, up, down, forward, back. Okay, the other critters like a sea anemone is round. It has radial symmetry. Radial symmetry means if you cut it in any axis, you'll cut it into two equal halves. We also have fossils a complete digestive tract. So we got a digestive tract that involves specialization. Parts of the digestive tract that acquire the food, the teeth, the mouth. Parts of it that masticate it, chew it up. Parts of it that digest it. Parts of it that get it ready for waste expulsion, right? And that goes hand in hand with segmentation. Because if you think about the earthworm again, if you segment it, then in different segments, different things can happen. It's like a conveyor belt at a factory, right? It's the same idea. All right. So in the Cambrian explosion, we get this world that looks somewhat on your left with the arthropods, the jointed organisms. Okay, sorry about that. Lost connection there for a second. Okay, so we're talking about the arthropods, this imposing looking thing, Animal Marcus there, which actually, if you look at it, the whole idea behind here, or the whole thing I'm trying to show is that's a pretty good looking mouth and food gathering appendages, right? So we have some very, I guess you could call them simple critters, although they were doing pretty well for back then. This one down here is another animal that we'll talk a little bit later on. That's actually a very cool critter with its repeated segments. In the background, I point out a chordate. You're related, us humans, we're related to chordates. So even back then, our ancestors were around, showing once again that it's not a ladder, right? Everybody kind of starts about the same time. They just go off on their different ways. Okay, reasons for the explosion again. Arms race, predator-prey relationship. Atmospheric oxygen rose, okay, because we have those photosynthesizers, higher metabolic rates with a higher O2, larger critters. Larger critters have higher metabolic rates. Higher metabolic rates, you got to have more oxygen. And then again, those Hox genes. That shuffling around of the Hox genes, that duplication that ended up with all these variabilities. So when we move out of the Ediacaran animals beyond the Cambrian explosion in what we call the Paleozoic, 450 million years ago or so, we get animals moving out on land. Insects first, okay? Insects would have moved out first because they would have moved out in conjunction with the plants, especially those early ones that were pollinators. Insects would have been the first ones to move out. They could have fed on the plants, then the things could have fed on the insects. And then we jump up a little bit faster, and we find out that, as we move from the Paleozoic into the dinosaur era, which is the Mesozoic, we see these animals out there that are doing something very weird. See those fish, those lobe-finned fish? There are lobe-finned fish that are still alive today. Most of them are extinct. But what we mean by a lobe-finned fish is we're talking about a fish with legs, basically. Not a complete leg. Remember, we talked about that in evolution. We'll get as 5% of a leg, but still a leg. And from lobe-finned fish, it's a small little change in that ladder there to get these early amphibians, to these critters that lived out on land but still had to go back to the water to lay eggs. Okay, we'll talk about those as we move through it. All right, and so now we're at the end of the Mesozoic right there. The dinosaurs go extinct. So, of course, now we're going to be moving into the era in which, we come on the scene. But, I mean, there was a lot that was going on in between that. The dinosaur era, of course, as I said, was the Mesozoic. I'm sorry, I kind of skipped this slide in my talk. I wanted to go into the Mesozoic. We get, in the Mesozoic, we get the dinosaurs. Yes, we get dinosaurs that move back into the water. These are plesiosaurs. They were dinosaurs that lived on land and moved back into the water. That's something we see in mammals, too. Marine mammals, whales and dolphins were mammals that lived on land at one point, and then they moved back into the water for resources again. The birds were coming around. They weren't flying yet, but they were, groups of the dinosaurs were experimenting with feathers because they were warm-blooded. We were around again, us mammals, if you will, but we were spending our time at night running around. We know this because fossils had big eyes, and that's a good indication of low light levels. And why were we coming out at night? Because the place was full of these damn dinosaurs, and they just loved to eat us little mammals. So being at night was a good way to survive. But, of course, dinosaurs ran for, 200 million years until the asteroid hit 65 million years ago, extincting the dinosaurs and lots of other things. We see, for one thing that happened in the lower left, that after this, the world got cooler. For one thing, the asteroid would have thrown all this stuff up in the air, which would have blocked out the sun, and that killed the plants, which eventually killed the dinosaurs. But it also would have made the Earth cooler. When the Earth gets cooler, it gets drier. So what you see in the Cenozoic there in the middle, the mammals, we would have seen mammals in a different environment, a cooler and drier environment, less trees, less forests. And that's where we come into the scene in the lower right. Now, I don't talk a lot about human evolution in this class because of the fact that that's a whole course unto itself. But one of the things we do know is during this time when the Earth was getting cooler and drying up and there were less forests, there were certain primate lineages that came out of the trees and began to move out into the open area. When you go out in the open area like that, one of the best ways to help yourself survive is to be able to see further than the predator. So stand up on your hind legs. So bipedal locomotion may have been, in some cases, a response to being able to stand up to see your prey. And stand up to see your predator. Something coming after you. Good defense, right? Okay, so that's our history lesson in a nutshell. Let's go back into this idea of what's so cool about animals. Well, we already talked about this a little bit. Symmetry, we'll talk about tissues a little more. We'll talk about body cavities that I introduced you to. Changes in the circulatory system. What it means to be a protostome and a deuterostome. Stome means mouth. Okay, proto is first and deutero is second, so I might give you a little hint. And then this idea of segmenting again, segmentation, or what we call metamerism, how important that is for the different diversity of life. But let's go through symmetry again real quick. If you're radially symmetrical, you can draw a line and divide into two halves. Now sponges are actually asymmetrical. All right, they don't really have asymmetry. Even the ones that kind of look like they do, they're still that loose aggregate. Radially symmetrical organisms are many times sessile, so they're sedentary, but oftentimes the larvae are planktonic. Since the larvae can't get away, then they drift away, right, for the reasons we were talking about. You were supposed to disperse. Now, if radially symmetrical does have an advantage, though, in the fact that the organism, they don't move very fast, but sea anemones, if you watch them sometimes, will move a little bit. And they can move in any direction. They can be aware of threats from any direction, right? They're like a castle, if you will. Down below, if you're bilaterally symmetrical, you're going to have your head in the front. It's a it's a characterization we call cephalization. It's over there on the right. It's putting the sense organs up front, which makes sense to us if you're bilaterally symmetrical, because we don't move in any direction. I know you can walk backwards, but the sea turtle, when it swims, is going to swim towards its head in that direction, right? And when you walk, if you're walking on all four, you would walk with your head like a dog in the front. You would want to sense your environment. So you at first, or initially. So you put the head in the front where it could easily tell you. It's kind of like standing up on your hind legs, isn't it? Put the head in the front so you know if you're coming up on prey, so you can eat it, or if you're coming up with something that wants to eat you. So once again, the fact that we see radially symmetrical animals today, like the jellyfish, means that doesn't mean that doesn't work completely. It works in certain environments. But most of the critters today have what we have, bilateral symmetry. They have an anterior, a posterior, a dorsal, a ventral, a chest region, a front, a back. Of course, the consistency of movement is also an advantage, right? When you're radially symmetrical, maybe you don't have to turn around to go in the other direction, but when you're bilaterally symmetrical, you can more easily stay consistently in that direction. So you can migrate, move, and attack things. Now, echinoderms are weird. The sea stars actually have both types of symmetry. They have radial symmetry when they're adults. It's actually got a special name. Let me get into the chat box here. It's called pentaradial. Fives, although not all sea stars are five, by the way. But when they're young, by the way, when it's a larvae, they have bilateral symmetry. And that's interesting because later on we're going to talk about which invertebrate group did the chordates spring off of. And you might be a little surprised, especially when you think about what they look like today. Okay, tissues, here we go. This has to do with stem cells too. Here in the upper right is a picture, one cell, two cell, a blastula, maybe more like a morula, then a blastula, there's a hollow cells. And then, of course, when gastrulation would start, what happens? We would pinch in at that point, right? Okay, an animal zygote is totipotent. Now, what that means by totipotent has to do with the cells. The word totipotent, the animal zygote, and the morula, there's that word morula, both have cells that can give rise to any tissue type. Now, totipotent is different than pluripotent, which you see in the red down below. Pluripotent cells can give rise to any tissue type in the body. Totipotent cells can give rise to any tissue types in the body and the tissue types necessary for the placenta. All right, so that's the difference. Totipotent cells can give rise to all parts of the placenta, the umbilical cord, and all the tissue interface that occurs, and the heart, circulatory, nervous system, immune system, whereas pluripotent cells can only give rise to those organ systems. Now, or cells that give rise to those organ systems. This is what we mean by stem cells. Stem cell research, if you think about it, if you have a problem with your circulatory system, your heart, if we could go back and use stem cells, we could grow another heart. That's the point behind it. And if we have some of your stem cells, we could grow another heart that would not be rejected by your body because it would not be seen as foreign. All right, and you notice what the situation is. Once you get to be in the circulatory system or nervous system, then you're unipotent. Then you can't go back, right? Heart cells can't be used to make a liver and so forth. So cells in animals are a lot more deterministic when they're formed than they are in other organisms. And of course, like I said, if you go into Bio 1, you'll learn that the idea behind this whole thing of cells and tissues is so important in animals because look, with cardiac tissue, different kinds of cardiac tissue that work in different ways, nervous tissue, epithelial tissue for your skin, things like that. Now, this doesn't apply to the parazoa. The parazoa are the peripherans, those sponges in the lower left. They have no defined tissues, therefore no organs. Can't have organs without tissues. Now, I guess an advantage if fragmentation in periphery is possible. What do you mean by that? You can take a sponge and grate it on a grater, and each little individual piece of the sponge will grow back into a full sponge. You couldn't do that with us, could you? You could take a cheese grater to your arms. I wouldn't recommend that, but you're not going to grow another human being out of that. It doesn't work that way. So parazoa, zoa means animal or tissues. Para means not quite tissues. Metazoa means true. EU is true. And meta is many. So well-defined tissues and organs, irreversible differentiation. Once they become unipotent, they can't go back. Parazoa can fragment because they don't have irreversible differentiation. If you cheese grate that sponge, even one little cell has all the information to make all of the other cells that are necessary. We'll see more that there are cells in sponges. And they do things, but they're not quite tissues. More tissues. Actually, tissues and body cavity. Okay, once again, let me introduce you to these Germanic. Yes, they're from Germany, Steve. Germ tissues: ectoderm, mesoderm, and endoderm. And again, in science, when we use the word germ, we refer to something that gives rise to something. That's why we use it for disease. It gives rise to the disease. Now, this isn't disease, but the ectoderm is the embryonic tissue layer, the germ layer that's going to give rise to things like our integument, our skin, and components of our nervous system. Mesoderm is eventually going to become those cells that's going to become tissues involved with things like the skeletal system and the muscular system. And the endoderm is going to be the innards, basically, the digestive organs, the intestines. And there's lots of things I left out. But you see what I'm saying here? That's what those layers are doing. All right, those different germ layers that are forming way back when I showed you the blastula are separating out into these three groups. Now, not all have three. If you have three, you're triploblastic up above in the red. But the cnidarians and the ctenophorans, the C is silent in those words, those are jellyfish and comb jellies, only have the ectoderm and the esoderm. They are diploblastic. All right? So they're going to be relatively simpler animals than us. Okay, now, those germ layers are important in developing the three types of body cavities in animals. All right, let's start with the easiest one first at the top. A body cavity is a coelo. Anything that's like blastocele, C-O-E-L, that is a cavity. So a coelomate, right, means that you have a body cavity. Now, in science, when we put an A in front of the word, like this one, or if the word starts with a vowel, we do this one. I didn't spell it right, but you get the idea. Right? Means the opposite of. So up at the top, that's a flatworm. We'll talk about those later. They have no body cavity. They are acoelomates. What do I mean by that? Well, when you slice them open, what you get. Now, I'm not talking about the cavity in the middle. That's the digestive cavity. By the way, folks, that's what the Archaneron became. You're pushing that hole through the balloon. That's what you made. Not the digestive cavity, but I'm talking about the space between the blue, the ectodermal derived tissue, and the yellow, the endodermal derived tissue. In between is the mesoderm, the one in the pink, and it's fully filled in. There's no body cavity there. Again, not the digestive cavity. All right? Now, if you jump down below to the pseudocoelomate, you do see a body cavity. Now, I'll talk in just a second about why it's called pseudo, which means fake, even though you're looking and you're going, I see a body cavity there. There is a body cavity there. And as I said before, body cavities are good for accommodating, supporting development of organ systems that can work together, that can be efficient. And body cavities can be used in some cases to make repetitive organ systems. So if one goes bad, then you still got the other body cavities organ system to take over for it. That's not a bad way to go about life, right? I wish I had like maybe a spare liver or something. Roundworms have a pseudocoelomate. Now, okay, again, the tissue is what makes the body cavity. Ectoderm, mesoderm, then you see the body cavity and you see the endoderm. And you can obviously see the body cavity, the pseudocoelom. Now, why that's called a fake coelom is a technicality. Obviously, there's a coelom there. If you're a coelom, meaning true, there we go. If you're a eucoelomate, then the difference is your body cavity is surrounded on both sides by mesoderm, both sides by the pink. All right, see the difference between the two? It's a technicality, right? Don't worry again about what the division at the top, the little lines at the top and the bottom are. We'll talk about that in just a second. But you see what I'm talking about? So both pseudocoelomates and coelomates have body cavities. It's just a technicality. If you're a true coelomate, then mesoderm lines both sides. Coelomate, the mesoderm only lines the outside. All right. Circulatory system. Hello, Marie. I'm sorry about that. I don't know what's going on here. I just totally lost connection. I couldn't get back on. All right, so any questions on body cavity, right? The difference between them, of course, is going to be the mesoderm. I said let's move on to the circulatory system. All right, there's two types of circulatory systems in organisms, two types that exist today. One's not better than the other. One is more, just works better where it is. The open circulatory system. All right, in the open circulatory system, we have a heart, we have a pump, we have vessels, but those vessels dump into big open areas in the body cavity called sinuses. And the blood actually mixes with the lymphatic fluid, the lymph that you have in your body to form something we call hemolymph. You see it in the picture there in the yellow. And the hemolymph is in these sinuses that basically surround the organs. So if you look at the picture of the grasshopper down below, the heart across the top pumps blood out these open-ended vessels. That's why it's called an open circulatory system. And the innards, the guts, if you will, just kind of float in this sea of blood and lymph, this hemolymph. Now, this is only going to be effective if you're small, very low metabolic rates. It's not going to work really well if you're bigger and you have a higher metabolic rate. That's why we don't have an open circulatory system. So things like clams and a lot of the insects, crayfish, are going to have this open circulatory system. That works. We have, of course, the closed circulatory system, as does the earthworm, right? Blood leaves the heart. Now, in our case, we have a more complicated heart. Blood goes to the lungs, then to the heart, then to the body, then to the lungs, right? That loop, double loop. But regardless, it's all closed. We never dump our blood into the body cavity that way. Now, mixing in with this whole idea of the circulatory system is what happens to the heart. As organisms get bigger, well, for one thing, it's physics. As organisms get taller, then it means you need more blood pressure to push the blood up to the brain. And of course, as organisms get bigger in general, you need a stronger pump. So the heart is going to get bigger as organisms get bigger as well. And if you're thinking, does that work in the open and the closed? Well, remember now, you're not going to have large open circulatory system critters, right? They only do well if they're relatively small. Okay, this whole idea of this unique embryonic development that occurs, and what's the difference between a protostome and a deuterostome. So let's take a look at this chart. Let's start with the first difference between protostomes and deuterostomes, cleavage, how the cells divide. In protostomes, you notice that when the cells divide from the 4 to the 8, the new cells, the ones in the blue, are twisted, right? They're turned. It's called spiral cleavage because, of course, it's spirally turned. Organisms that are deuterostomes, it's just much simpler. 4 cells, the other new ones happen right on top. We call it radial cleavage. Now, this is interesting too, protostomes and deuterostomes in terms of what we call the fate of the embryonic cell. So in other words, back at that 4-cell stage, is their path already charted? Is their development determined? If you're a protostome, yes, it is at that 4-cell stage. That means if you take one of those 4 out, okay, then you're not going to get the embryo. Development is arrested. You've taken out some crucial information. But back to this whole idea of being totipotent and pluripotent and animal cells in deuterostomes, in many cases, or deuterostomes, the 4 cells have indeterminate development. So you take one out, no big deal. The information is then exercised by the other cells. It's how you get identical twins, folks. We wouldn't be able to get identical twins if we had determinate development. Indeterminate development, in human beings means that the zygote splits into 2 separate entities. And since each has the same information and it's indeterminate development, you're still going to get 2 guys. That's not really a great picture of identical twins, but you get the idea. All right, that first indentation, if you're a protostome, the blastopore, right, becomes the mouth. Protostome means first mouth. If you're a deuterostome, guess what? Your digestive system starts with the anus, so it goes the other way. So deuterostomes, humans, by the way, we're a member of this group. The anus there starts from the blastopore, and then it becomes the mouth. All right, formation of the coelom. In other words, where does the body cavity come from? So now we're going to be specifically just looking at eucoelomates here, or coelomates, those that have the true body cavities. In the case of a protostome, the mesoderm, the pink tissue, starts from cells that are floating around in the blastocele, whereas in the deuterostome, the mesoderm starts from outpocketive cells of the endoderm. Either way, what those cells will do is they grow and form the lining of the body cavity, which, as I said, is what constitutes them being a true coelomate, right? Because they have the mesoderm on both sides. Pseudocoelomate wouldn't have the mesoderm around the inside. It would only have it around the outside, remember. Okay. Questions on that? Did the recording start again? Yeah, it did. Sometimes the recording doesn't start again after I do that. All right, and more about this, especially when we get into the insects, but this is what I mean again by segmentation. That's another very interesting innovation of animals. They begin to take these Hox genes, right, with their shuffling and moving them around and so on and so forth, and begin to make different areas of the body. Like if you look at this, very generalized, I don't know, lobster, shrimp, crustacean up above, you can see the segments. We're counting them. And if you think about a lobster or shrimp, the tail especially, right, that's segmentation. But we could have the cephalic segments. We could have the cephalic segments combining with the thoracic or the chest segments, the cephalothorax. And again, it's not so much that it's divided up, but the fact that it's divided up and it's specialized. You notice like the cephalothorax has got the antenna on it. It's got the walking legs. The abdomen has got the swimming legs. All right. So what we're doing is specializing throughout the organism. We get this not only the ability to have different body parts in different areas, but having segmentation also means you like the guys in the suits of armor, right? You can move. You got flexibility. You can also make more than one, like I said. Like the earthworm has a kidney functioning thing. It's not really called a kidney, but they have an excretory system in every segment of their body. So if this one goes bad, doesn't work, no big deal. It's not like losing a kidney. They've got several kidneys. Segmentation, we have it too, right? Look at the picture of the x-ray. Look at our vertebrae, our ribs. It's all segmented, okay? So we're not immune, or not immune, but we're not different in any way that. Now, this whole idea of segmentation doesn't mean relationship, okay? Segmentation is obviously a genetic characteristic, one of those Darwinian advantageous variations that many different groups of animals came up with, right? So because you're segmented doesn't mean that you're closely related to any of this. As a matter of fact, I already kind of hinted to this. If you think about the echinoderms again, sea stars, sea urchins, sand dollars, we're more closely related to them than we are to anything that's segmented. And sea stars and sand dollars aren't segmented. Okay, so segmentation is it's not monophyletic, right? Doesn't mean you're related to one another. Oh, I got perfect timing here. So, last slide. This is where we're going to go from here. This is the last group of critters we're going to look at. Obviously, we've talked about our protozoan roots, right? And so now we talking about a group of organisms we call the metazoa. Metazoa just literally means multicellular. Eumetazoa means tissue multicellular, right? So that's why the poriferans and are in the metazoa, whereas the ctenophorans and the cnidarians, and again, these words will become a little more familiar to you, the comb jellies and the jellyfish are the eumetazoa, because they only have two. They're diploblastic, but they have the ectoderm and the endoderm. Okay, now, two tissues, of course, they're diploblastic. Now, some of these groups we won't talk about, or we'll just briefly talk about, like the Acela and the Hemichordata, I don't really talk about. I'm going to skip the Deuterostomia for a second. So we move up now about 670 million years ago, and organisms are developing the ability to move from symmetry, that's radial symmetry, all right, back to a situation where they have bilateral symmetry. Now, if you're looking at the sea star here going, wait a minute, that doesn't have it. Remember I said the larvae of the sea star is bilaterally symmetrical. And that says something very interesting about a relationship between us and those guys. All right, so the bilateral organisms, and skip the Deuterostomia for a second, move down here, these two groups, which are broken down by what type of larvae they produce. The Lophotrochozoa, right, produce a larvae that's either looks like this lophophore, And Lophophore is members of a group right now, the Ectoprocta, but that's the larvae they make. It's a filter feeder. Or they make the trochophore larvae. So again, similarity of larval form is combining these animals together. But together, the Lophotrochozoa, and I love this word, the ectozoa. Ectozoa, ecdiosis is to molt, like that blue crab, when organisms molt. Nematodes do, but you probably know more insects and crustaceans that do that. Both of those together, the Lophotrochozoa and the ectozoa, those are the protostomes. So they form their mouth first during development. And we'll go through each one of those. We'll go through the platyhelminthes, the flatworms. Don't really do the Ectoprocta or the brachiopoda, mollusks, annelids, nematodes, arthropods. The deuterostomes then, very small group, involves our closest cousin in the invertebrates, the echinoderms, and then here we are. We are the chordates, and we're triploblastic, right? 3 layers of tissue. Okay. Any questions on any of that? That's it for chapter 32. What we'll do now in chapter 33 is go on to the invertebrates, and then chapter 34 we'll finish up with the chordates. So if you don't have any questions, then that's all I have for class today. Let me know if you have any other questions by emailing me. Announcements. Exam Monday, right? Chapters 29 and 30. Your field notebooks are due next week on Tuesday? Is that right? Yeah. It's Monday. Yeah. So make sure you get those ready. And that's it, I think, for now. Okay, so I'll see you tomorrow. We'll be doing dissections. All right. Take care.